Okay, let's go ahead and get started, everyone. So it's my great pleasure to introduce Professor Sven Dickinson. Sven is our esteemed colleague in the Department of Computer Science. He's a long-term member of the computer vision community. He's a previous department chair in our department. He's been editor-in-chief at me, and he's also recently the former director director of the Samsung AI Lab, which many of you know. And he's also done great research in understanding shape. He's recently interested in human visual system, and his research has been recognized by Premier's Excellence Award. He received a Lifetime Achievement Award from the Canadian Image Processing and Pattern Recognition Society. And so, yeah, excited to hear from Sven today. Please join me in giving him a warm welcome.
Thank you for the kind introduction, David. I'd like to thank David, Avia, and Marcus, the organizers of the Toronto Vision Seminar, for their kind invitation. I think it's been about 26 years since I've given a talk in this department. That's when I last interviewed here. So it's a real treat to come back and give another talk. I'd also like to thank Jasmine for helping to set up everything. She's been terrific. And of course, all of you here in person and all of you online for joining. I really appreciate that. Before I begin, just a little context. David mentioned that I used to be department chair. I finished up about 10 years ago, and I went on my sabbatical, and I decided to pivot towards something that I've always had a deep passion for, and that is shape perception and human vision and how it can better inform computer vision. So I hired a psychology postdoc, I assembled a team of interdisciplinary researchers spanning both human and computer vision, and started on a new research program. But 2 years into it, I got distracted by the Samsung job that David talked about. And while that took some of the wind out of my sails on this work, I was fortunate to work with some wonderful collaborators who kept the momentum going. And so I'm recently back. Last summer, I retired from Samsung. I'm back in the department 100%. I can't tell you how happy I am to be back, reconnecting with my colleagues here. This is my first love and my first home. So it's wonderful to be back, and it's especially wonderful to get back on this research horse that I started on about 10 years ago. And what I'm going to talk to you about today is some of the progress that we've made over those 10 years, less than that, I should say, and some of the things that lay ahead, some of the things that we're planning to do. So with that, let me begin. But before I dive into my work, I'd like to dedicate this talk to a dear friend and colleague of mine, one of the world's foremost researchers in human vision, focusing specifically on saccades, eye movements, and attention, Eileen Cowler. Before I was a professor at Rutgers, my wife Suzanne and I were both professor at U of T. My wife and I were both faculty at Rutgers. When we started our faculty jobs, we benefited from the enormous friendship and mentorship of Eileen Cowler, who is a professor in psychology there. I'm sure those of you who have gone to a new place can appreciate what it's like to have one or two people to help break the ice for you, to make you feel welcome, to help you settle in your family and understand the landscape that you've just joined. Eileen was one of those people for me. I dearly miss her. She was a wonderful mentor and teacher to her students. And so again, I'd like to devote this talk to her, dedicate this talk to her. So this talk is about symmetry. As you know, symmetry is ubiquitous in nature. Look around the top row, I've shown you examples of bilateral and rotational symmetry in the animal kingdom, the birds, cats, shells, peacock. On the middle row, I've shown examples of symmetry, both reflection and rotation in the plant world. Here's one from the natural world, an ice crystal, a snow crystal. If you zoom in down to the microscopic level of bacteria or microbes, you'll see all kinds of symmetry. If you zoom out into the heavens, you'll also see symmetry. And this one I added just for Aviad. This is a black hole which exhibits spherical symmetry. I hope I got it right, Aviad, because I know you're listening. Now, despite the prevalence of that kind of global symmetry, in fact, local symmetry is much more ubiquitous in the world. Because even though an animal or a plant or whatever may have global symmetry, when it articulates or when your viewpoint it changes, that global symmetry is disrupted, but the local symmetry remains invariant. So you can see examples here where these animals, these objects may have global symmetry, but due to articulation and viewpoint change, especially you see on the bottom, both humans and cats have global symmetry, but when they articulate their joints, or you view them from a different location, you project the figure onto the image, it no longer has global symmetry, but the local symmetry remains invariant. So in fact, local symmetry in our world has shaped the evolution of our human visual system. The most compelling example that initiated me down this path happened in 1984. A year before I went to graduate school, I was in a bookstore and I bought David Lowe's book, which was a reprint of his thesis. And in the introduction, he had this figure. And it jumped out at me just as I'm sure it jumps out of you. So there are three non-accidentally related groups in this picture. I'm guessing you see them. Here they are. One is cotermination on the upper left. One is collinearity. And this one is symmetry, right? These are groups which the human visual system has evolved to detect as happening non-accidentally. If lines are close together and symmetric, there's a very high probability that they belong to the same object. They're not some random objects that are randomly oriented in the world, right? So the human visual system has evolved to detect these kinds of regularities and make inferences on them.
Can you take the ellipses off again?
Sure. You see it?
No,
Okay, so it turns out that local symmetry has a long history in the computer vision community. Probably the first example could be traced back to Harry Blum, who introduced what we call the medial axis transform back in the late 60s. The way it works is you have a silhouette of a shape, and what you see on the right is the locus of maximally inscribed disks that are bitangent to the contour of the shape. And if you trace out the centers of those disks, as you see in the green points, you will get what they call a skeleton or a medial axis. The nice thing about the skeleton is it captures the structure of the object. If you articulate the legs, the skeleton structure does not change, right? So it's a nice part decomposition that's quite natural, and it's based on symmetry introduced in 1967 by Harry Blum. Remember this because we're going to come back to it later on. In fact, there's evidence for local symmetry in the human visual system. When I was at Rutgers in the early 90s, three of my colleagues, Ilona Kovacs, Bela Ulesh, and Akash Fair, showed that contrast sensitivity increases in the vicinity of a medial point. In other words, if you're looking here, it's the center of a rotational symmetry, you're more likely to recognize that there's a change in brightness from here to here. So there's evidence for medial representations in the human visual system, as shown by these researchers. In fact, flipping back to the computer vision community, there's a very famous computer vision researcher from Stanford, Tom Binford, who introduced what we call the generalized cylinder. And in fact, it's arguably a medial axis in 3D. A 3D space curve, you sweep a cross-section through it. There's a cross-section function that alters the shape or scale of the cross-section as you sweep it. Neuroscientist David Marr, who you may have heard of, and his student Keith Nishihara talked about composing medial parts, 3D generalized cylinders, into more complex objects and argued for coarse-defined representations of these objects. And in fact showed, as you see on the bottom right here, that even with a single part category, namely a cylinder, a straight homogeneous right cylinder, impoverished as it is, when you attach it together in different ways with different lengths, different angles, and different attachment points, you can flesh out quite a rich vocabulary of objects. And in fact, the psychologist Irv Biederman, who we sadly lost a few years back, took the idea of a generalized cylinder with continuous properties and mapped them to discrete dichotomous and trichotomous properties: straight versus curve, cross-section, straight versus curve, axis, straight versus curve, etc. And he created what we call geons, which are again a symmetry-based qualitative representation, which he showed humans could recognize objects very quickly based on the qualitative symmetries of the part models. And in fact, if you fast forward about 40 years and hundreds, literally hundreds of papers on symmetric part-based object recognition, you still have researchers working on the problem today. How to reconstruct objects, making assumptions of symmetry-based parts, shape representation learning, and so forth is an active area of research today. Yet despite the fact that symmetry has long been the basis for object recognition, object representation, object reconstruction, and shape representation learning, the role of symmetry in scene perception has not been given much attention. And that's really the topic that I'm going to talk about today, which brings me to the two parts of my talk. The first part, we're going to pose the question, can we use our knowledge of symmetry in computer vision to help understand the role of local symmetry in human vision? And if we can find results and insight into how humans use symmetry, how do we take that knowledge, bring it back into computer vision, and inform the design of more effective computer vision systems? Okay, part one, role of local symmetry in human scene perception. So one of the wonderful collaborators that I teamed up with about 10 years ago is a professor in our psychology department here at U of T, Dirk Bernhard Walter. And in some earlier work, before we crossed paths, he showed that if you show photographs of scenes to humans, they're able to categorize them in under 45 milliseconds, and sometimes as quickly as 11 milliseconds. Moreover, when he did an fMRI analysis of these human subjects in a scanner and looked at their voxel activation patterns in different parts of the visual pathway, he found that there are particular areas which likely contribute to object categorization in the human visual system. So really the question we wanted to ask is, what is the information? What's the important information being encoded in these scenes that allows you to recognize the scene in 11 milliseconds, right? That's the big question. So what Dirk did was he hired human artists, and the human artists took his data set and they traced out the shape contours in the data set. Then he ran those same experiments with human subjects using the contours, throwing out all of the color and texture that was in the original photographs. And it turns out humans did almost as well, suggesting that shape, which many of us believed, shape is really the fundamental cue for object and scene categorization. So now the question is, now that we established that shape is extremely important, what are the features of shape that are relevant for categorization? So what Dirk did next in work in 2014 was he took the contours that the artist had traced out, came up with a piecewise linear approximation of those contours, and then computed histograms of both unary and binary features on those contour fragments. Orientation histogram, length histogram, curvature, the angle between two adjacent pieces, junction type, as you can see there, and junction angle. Then he trained classifiers to be able to predict what the scene is given the particular histogram of features. And what he found is that there was, if you correlate the errors that the classifiers made with the errors that humans made, he found that there was little error correlation for unary features, namely orientation and length. However, there was very high correlation among the binary features or the relational features. This is no surprise to someone who's worked in perceptual grouping. I showed you those generalized cylinders that Mara Nishihara proposed. The relational information is extremely powerful, much more powerful than the unary information, right? So now we have this hint that it's the relational information among these contour fragments which somehow plays a key role in object categorization. So what was our next exploration? So at the time, there's been competing theories in object recognition about the relative importance of junctions, which are a relational information of two lines that meet at a point, right? Versus the middle segments, right? Which have a different kind of relational information. So what we did was we took our line drawings and we created two stimuli. We took each line and we created two stimuli, each containing 50% of the contours. In one case, we remove either the junctions. In the other case, we remove the middle segment. So let me show you an example which will make it very clear. Here's the intact line drawing. Here's removing the outer 50% of each line. That's the junction. Here's removing the inner 50% of each line. These are the middle segments. The union of the two gives you the intact contour figure. So here's the intact line drawing for one of our scenes. Here's the result of removing the junctions. Here's the result of removing the middle segments. First quiz for you sitting in the room is what is easier to recognize for you? Is it this figure or this figure? Removing the junctions, does that make recognition easier? Or removing the middle segments? Hands up if this is easier. And hands up if this is easier.
Looks messier.
Yeah. Well, the first group had it by majority, right? Removing the middle segments hurts recognition the most. You can see the results of intact versus junction remove, middle segments retain, and middle remove, junctions retain. So it's a clear advantage to these middle segments. Why? What's the hypothesis behind this? Here's what we're thinking. In order to understand the scene, you have to extract the 3D structure, 3D surfaces in the world. 3D surfaces project into 2D closed contours in the image. Right? So every surface in the world projects to a closed contour in the image. If you remove 50% of the contours, you make contour closure hard in a scene, in a complex scene. If you remove the middle segments, you remove the non-accidental longer range symmetry relations that help our visual system close these contours. So how do we test this hypothesis? Well, as you saw in the previous experiment, we just deleted each half of every segment. The outer 50% or the inner 50% had nothing to do with symmetry, nothing. So what we need instead now is a score, a symmetry score for each contour pixel in the image that measures the extent to which it participates in a non-accidental symmetry relation with some other contour pixel in the image. Once we have such a scoring mechanism, we can go back and create two stimuli again. One, top 50% scoring symmetry pixels. The other, the bottom 50% scoring symmetry pixels. The union of them giving you the intact contour image. How do we compute a symmetry score for every contour pixel in the image? Here's where we go back to Blum's medial axis transform, which you recall is the locus of maximally inscribed disks that sweeps out the shape. The green points are the medial points, and you can see the maximal circles there. So how are we going to do this? Well, let's take two contours in the image, C1 and C2. We're going to have a point P on one of those contours. We need a score, a symmetry score for point P. That's what we're going to sort on at the end of the day. What we see in blue is the medial axis computed for these two contours, C1 and C2. In fact, point P is tangent to a maximally inscribed disk that's centered at medial point Q naught on the blue contour. Now we can step left and right to create a window of medial points centered at that location. And what we simply do is look at how the radius changes along that window. And we can come up with a very simple expression that looks at how often the radius changes small enough, normalized by the size of the radius, to give it a parallelism like this, right? So we have a score that really reflects local parallelism at a given medial point. And that gets assigned to point P. The only problem is, we don't know that C1 and C2 bounded the shape. Could be the outside, not the inside of a shape. And in fact, point P also defines a medial point T naught on a different medial axis. And so what we simply do in the end is we choose the maximum of the two scores, right? So what we have now, for example, just for illustration, we have our original image. We have our line drawing that the artist created. We have a simple distance transform. The work of my collaborator, Khalid Siddiqui at McGill, computes a local feature called average outward flux. That gets thresholded to form skeletons. We can do a connected components analysis on those skeletal fragments. We can score those according to our symmetry score, and we can sort them, top half versus bottom half. So quiz 2, hands up if you think that the left one is easier to recognize. Hands up if you think the right one is easier to recognize. All right, you all win. All right. So the effect is even more compelling. So here's some examples. Here's one of our categories: beach scene. This is the intact scene. This is symmetry on, back to intact, symmetry off or low. Here's a cityscape scene, high symmetry, back to the intact, low symmetry. How did we do the experiment? We showed subjects one of the stimuli for about 53 milliseconds. Then we flush the visual pathway with random lines, noise, if you will. Then we give them as long as they need to enter a forced choice response on what category they're looking at. And then we repeat for about 300 trials. And the results are even more compelling than the last set of results. Symmetry has a clear advantage. It encodes extremely important information in support of human scene perception. So what was next? We took our...
Can I ask a question about this?
Sure.
So this encodes like one particular type. You're analyzing a particular type of symmetry, right?
Yes.
Like
Yes.
parallel lines?
Yes, I'll get to other kinds of symmetry
okay.
later.
All right, all right.
Yeah, very good. You're ahead of me. So what we did next was we took our subjects and we put them into the fMRI scanner, which we have here in psychology. And we're able to partition the fMRI image into the 8 components of the visual pathway, beginning with V1 and up to RSC. For each chunk of voxels, we train a classifier to be able to predict what the human is looking at by looking at the voxel activation in their brain in that particular region of their brain. We don't need them to answer any questions. We have ground truth. We know what they're looking at. The question is, can we predict what they're looking at from these different regions of the brain? So what you see in the results, the dashed line is chance. They all do better than chance, right? But if you look at area PPA, which in fact is the area that human vision researchers believe is responsible for scene categorization, not only do you get significantly better performance than the other regions in the visual pathway, but you get the same symmetry, non-symmetry differential that we saw in the behavioral studies, which really suggests that area PPA is utilizing symmetry information to be able to perform scene perception. So what's next? What's next in this direction? Well, you recall our closure hypothesis. Let me repeat. Removing the middle segments removes the non-accidental symmetry relations that help our visual system close contours. We've established only the perceptual advantage offered by the presence of symmetry for scene perception, but we haven't drilled down and established the presence of symmetry for improved closure perception or closure performance, right? So what we're currently doing, I don't have results to share with you yet. What we're currently doing is focusing or narrowing in on the closure problem, which is one of the most important problems in human scene perception, human vision, frankly, is how to close contours. And we believe that symmetry plays a really important role in that process of closing contours. Okay, so that's what's next in that direction. Now let's go in the other direction. Now that we understand the importance of symmetry in human scene perception, how can we leverage this to develop better computer vision systems? The first question we asked is, does a CNN behave like a human? Well, the problem is, to begin with, we can't train any CNN with only 475 images in our artist line drawing data set. It's clearly not nearly enough data. So we need to collect some additional data somehow. So we went to the MIT 67 data set, which had about 6,700 images, and also to the Places 365, which had about 1.8 million images. Now, the problem is we don't have enough money or time to hire the human artist to trace out millions of images, right? Yes, Tony.
But I wasn't trained on line drawings. I can still read it.
Well, let me say that. That's a great open question. I'm going to get to that, right?
Okay.
Let me get to that. Great question. So we need a way to automate the extraction of these contours from these images, right? So what we did is we took a very well-known method of edge detection from Steven Zucker and Lee Iverson from McGill many years ago, tweaked it a little bit. We got something that performed really well. And in fact, we got a contour extractor that gave us about 90% overlap with the human annotated contours, right? So it's not perfect, but maybe it'll get us somewhere in the ballpark that we need. And it was also an opportunity, and this gets to your question, David, an opportunity to move beyond parallelism, which we call ribbon symmetry, towards taper symmetry, right? Why? Because any parallel symmetry in 3D will never appear parallel in the image unless you're viewing it orthogonally. All right, so any railroad tracks are clearly symmetric. They look like they're tapered in the image, right? So it's a much more powerful form of symmetry if we can somehow bring taper into our saliency-based score. Since we have medial radius computed at every medial point, we can look at the role of scale or size, right, as we see here, separation. And in fact, if we look at the local curvature of the symmetry axis, we can measure curvature. We can measure a notion of local mirror symmetry, right? The more curved it is, the less mirror symmetry. So now we have 4 measures that we can compute. Here's a demonstration of the heat maps for all 4 measures. Red is high, blue is low, and in fact, just as we did before, we can create this 50-50 splits using our saliency-based score. So you can see at the top, we have the bottom 50% score, half of the pixels. Next, we have the top 50%, and if you add them together, you get the fully intact original line drawings. Okay, so now we have more stimuli. So, first thing we did is we went back with our human subjects, and we tested them on the artist scenes with all 4 scores. Not just the ribbon symmetry, but all 4 scores: taper, separation, and mirror. And it turns out, not surprisingly, they had the same behavior with parallelism, ribbon symmetry, but they showed the same preference for the top 50% for these other saliency measures. We have the same differential for all of these forms of symmetry-based or medial axis-based saliency. What happens when we train a CNN to do the same thing now? Remarkably, at least to me, the CNN showed the exact same behavior with symmetry on, symmetry off, taper on, taper off, etc. for all 4 conditions. And I should note that neither the humans nor the CNNs ever saw one of the splits at training time. The splits were only shown at test time. Yet they exhibit the same behavior, the same preference for symmetry that humans did, suggesting that somehow the role of symmetry is equally powerful for a CNN to leverage in support of the task of scene categorization. So now the question is, can a CNN leverage these cues somehow? So let's review our pipeline. We start with a photograph, upper left. We move to our line drawing. We compute our medial axis. Trace out, we threshold out the medial axis fragments. Compute the connected components. You can see them here, different colors. Then we compute our 4 medial axis-based saliency scores. I'm showing just one here, parallelism. That gives us the parallelism scores on the MAT. Then we migrate those scores to the boundary pixels, the actual contour pixels, in the manner that I showed you, right? So now we have parallelism scores on contours. And now what? Well, our baseline condition for the CNN will be to take the contours, just the contours, without the scores, and stuff them into all 3 channels of the CNN. This is the baseline. But we're going to compare that to stuffing the contours in one channel and then loading up some of these saliency values in these other channels. And we can look at all possible combinations of bringing these extra features into the CNN. Here's what we got for the places 365. You can see in the dotted line here is the contours only. That's the baseline performance. And what you see is, by adding these additional scores, you can boost performance as much as 12% over the baseline of contours. Now, we tried a number of control conditions, adding local curvature, adding local intensity, gradient magnitude. We even randomized the saliency values on the contours. None of these control conditions matched the raw contour baseline condition. So somehow, the CNN is not able to fully leverage the symmetry-based information that's in the images and clearly benefits from our explicitly adding that information. So what's next? What's next along this front? As I said, the networks we train do not appear to be able to take full advantage of this human vision prior of symmetry. What's missing? How do we better leverage what we know from human vision to design better networks? You could take a traditional model-centric approach. It takes a fixed benchmark, which is a data set and a task, and you iterate on the design and structure of the models to optimize task performance. In such a scheme, your human vision priors could be encoded as handcrafted architectures or handcrafted loss functions or both, right? One example of how we might do that draws on some work we did a few years back, which established the state-of-the-art end-to-end pipeline for medial axis extraction from an RGB scene, RGB image, as you can see here, called deep flux. We could create an architecture that takes our image, jams it through this architecture. We could learn how to compute the saliency measures, given that we have a medial axis along the lines that I showed you. And then we could jam that into the input of a scene categorizer to see, is this the way to optimize performance on a scene categorization benchmark? We're not inclined to take that approach. Instead, we're inclined to take a more data-centric approach, whereby you fix the architecture and you systematically improve the quality of the training data to get the kind of results that you're looking at. So the key here is, instead of just choosing ImageNet or your favorite benchmark du jour, right? The key is to use your knowledge of these known human vision priors and ask yourself, how do we create meaningful training data that will elicit these priors as behaviors in your network? Because, Tony, to answer your question, none of these benchmarks remotely resembles how the human vision system evolves, right? We did not evolve to look at images, ImageNet or otherwise. We evolved to move around the world, to navigate, to grab things, to interact with the world. The world is constantly changing, as shown by one of our vision candidates last week. So, if we can come up with this kind of training data, maybe we can steer these generic networks into the right direction to be able to capture and reflect, which in our case which would mean showing it examples of symmetry in the world somehow, so that you tease out this implicit feature that humans have. Now, there's a whole community emerging in the human vision community that's taking off-the-shelf networks from the computer vision community and then testing them on human behavioral data. And a lot of people that are doing that are unfortunately quite negative, saying it doesn't match human performance. But there's no reason to throw out the baby with the bathwater here. I think the key is, the reason I believe these networks do not mimic human behavior is because they're trained on data that does not bear any resemblance at all to how the human vision system evolved over millions of years. Right? So, with that, let me once again say how grateful I am to my wonderful collaborators who carried this work while I was busy at Samsung, including Alan Jepson, who recently retired, both from the department and Samsung, Kaleem Siddiqui, John Wilder, my psychologist, Dirk, and some of his students. And we have a new team going forward. On the left is Zygmunt Pislow, a shape perception, cognitive scientist and human vision researcher at UC Irvine in the cognitive science department. Next is Dirk Bernhard Walter. He's the one here in psychology. Next is a new postdoc that's joining us in September, Matthias Tangemann, who's wrapping up his PhD at the University of Tübingen in Germany under the direction of Matthias Betke. Next is Tristan Omontado Armstrong, my PhD student who graduated working at Samsung, more or less a consultant on the project. And finally, we have Kaleem Siddiqui, my longtime collaborator from McGill University. All of these people are deeply interested in shape perception at the intersection of human and computer vision. And with that, I thank you, and I'm happy to take any questions.
Great, thanks very much. Questions? Yeah, Tony, Sai.
Your very last statement, you said one pixel.
Yeah. Yeah. I think that's a good example.
So it's trained on. So the main difference seems to be that you all were trained on video, not still.
Oh, well, we were trained in a dynamic environment with tasks, right? Now, sure, there's a bottom-up task. You blindfold a person, you take it off, they look around, they notice things, they form a model of the world, right? And that is a task that has to be solved, but there are many other tasks. Navigation, grabbing things for food, avoiding predators and so forth, interacting with other people, manipulating the world. There are many, many tasks that have led to the evolution of the human visual system, to the point where it does not make those kinds of adversarial mistakes. Now, there are visual illusions that it does make mistakes on, grant you, right? There are different class of mistakes than I think what you're referring to.
But that still begs the question. What's always evolution? What kind of visual system did it create? How's that?
What kind of visual system?
We don't have CNNs in our heads.
I'm not going to make that claim. It models a certain structure in the head. I don't want to. That's a kind of a religious debate. I don't want to get into, right? I believe that CNNs, other kinds of deep networks, have much to add, and they're very powerful tools. And I think we should not be ignoring them. But I think we need to look at more natural and realistic training conditions that match human evolution if we're going to use them and adopt them as models for human vision.
Okay, so Sai, and then we have a question online from Aviad, so we'll go to you, Aviad, after that, and then Kiros. And they're asking online if you could just repeat the question. So
of course, yes. Sorry about that. Yes.
So the experiment where you trained on the contours, I was just curious, like, did you ever try like just taking like where you were trained on like full images and just pass the contours to that? Because I guess that's how we are trained. Like I've not seen contours, right? Like did that just kind of fail completely?
So I think you're asking me, did we train on full images, including the contours, or past the contours?
Just
train on
just the images and then pass the contours.
So the networks, some of the networks that we trained were pre-trained on ImageNet with just the final 3 layers fine-tuned on the contour data. For the network I showed you at the end, in fact, we trained that from scratch, all layers of the network, but we found that the best network that we trained did not differ appreciably from the network trained on ImageNet.
Okay, and then one other quick thing. Have you looked into like temporal symmetry? Like I think this is like quite interesting where like you have like local symmetry but through time as well, and things kind of move like this.
Yes, glide or translational symmetry. Yes, we haven't addressed that, but it is a very powerful form of symmetry. You're absolutely right.
Okay.
Yeah, sorry, they asked whether we've looked at temporal symmetry. Aviad, did you like my black hole, Aviad?
Yeah, thanks for the nod. Yeah, thanks, Sven. I really enjoyed the talk, and I had maybe 2 questions if I have the time. So the first question is, at some slide you show the difference between the human visual system and the CNN when you feed different symmetries, maybe like 3 or 4 different symmetries. And one thing that I saw is that the human visual system seems to perform the same. Like there's an improvement in performance, but it doesn't depend on which type of symmetry. And the CNN seems to prefer certain types of symmetry. It seems like some symmetries give you a higher boost than the others. Is that something that you saw, and is that something that you can maybe comment about?
I need to find that slide, Aviad.
Yeah, this slide.
This one?
Yeah, so here it seems that the performance for humans doesn't depend whether it's taper, separation, or mirror, but it seems that for the VGG, for example, you see a much better performance for, I guess, separation and mirror.
Yeah, I can't comment on why. My own theory about why separation is really important is because when you look at the 50% high separation, you're looking at the larger, more salient objects in the image. It's a scale. It's a scale bias, right? Which is arguably not really a symmetry measure. It's a convenient feature that emerged from our medial axis point. It's a radius-based feature, a separation, right? But I think it reflects a scale bias in the image. And we know that large things can draw humans' attention, and maybe that would better support object scene categorization, right?
Gotcha.
Does that make sense?
Yeah, that made sense. And
I think the fall off on the MIT 67, we're looking at top 1, and we're looking at far greater categories than we saw in the artist scenes, which was only 6 categories, for example. So I think the more you go to a larger database in the top 1 measure, the more fall off you're going to get from intact down to symmetry, but you still have this significant differential between symmetry on and symmetry off. For all of
Yeah. Okay, and then that makes sense to me. The other question that I had is, you talked a little bit about the model-centric versus the data-centric approach. I think this may be a confusion in my head, but in my head, we as humans, we have the evolution of let's say, millions of years that constructed the visual system and the cortex and all of that. And then we also have from infancy, right? From the time that we were born, and maybe these two map to model versus data. And so why do we need to orthogonalize these? Why shouldn't it be, in parallel?
Yeah, I agree with you. The example I showed here, in my mind, is highly handcrafted, right? Based on a particular set of features and a particular representation that we have developed for object recognition and other problems, right? There is some kind of generic structure in the brain that has evolved over time, but there is fine tuning of certain functions from birth up to how many months the developmental psychologists say that the babies are learning these kinds of things. So I do agree with you that it has to be a combination of both mechanisms, right? We need some idea of what that foundational structure is, right? Ideally reflects the structure of the brain. And then we need to sort of fine tune that with real tasks, right? Does that help, Abia?
Yeah, that makes sense. Yeah. Thanks a lot.
Right. So I guess when thinking about human vision, I guess I mean, we don't look at photos the way cameras look at photos, right? We have a fovea, we have periphery, we have fixation, we have attention. So how could you say something about how you believe that plays into all of this? Like, the models that you consider, whether it's CNNs or the way these correspondences were established, don't really seem to take that into account. And I'm wondering how crucial you think it is for that.
So there's evidence that in the primate vision system you have more of a log polar mapping, right?
Right.
There's no reason we can't build that kind of architecture or use that as a default architecture. In fact, my colleague Zig Pisla, one of the collaborators, has written a beautiful paper on how closure in a log polar transformation, formulated as a search through cycles, which is exponential, how if you map that to a log polar representation is polynomial, right? Using a TSP. So there are many ways of massaging this base network, whatever it is, into something that better reflects the structure of what we know about the human vision system. I'm not a physiologist, one step at a time, right?
And the other related question I get, well, some of them.
But you're right. These are all examples of differences between the CNNs and the human vision system. Presumably, you want to bring them as close as you can along all these dimensions. I agree with you.
Have you thought at all about how one could maybe analyze like foundation models, let's say SAM, for sort of try to understand, like, is there anything tangible and any sort of perceptual correspondence?
That's something we could do. I have not thought about that. But I suspect that we're going to be exploring all these kinds of models that are popular today to see which of them would respond to the kind of training data that better mimics human evolution training data and delivers the human priors that we have found, along with other human priors, right? I don't think we're married to any specific architecture.
Oh, that's a question.
Yes,
so,
please.
I'm wondering about. So one interesting thing about CNNs is that they rely a lot on local texture. Right? So if you have an image of a cat and you took like the paw and the head and you scrambled all the region, all the parts, whatever, it'd still tell you it's a cat, even though it's, not really a cat anymore. And I'm wondering if looking at not just local symmetries, but like global symmetries or coarser scale symmetries could be useful. And is that Like, is that something you look at as well, or hierarchies of symmetry?
It's a great idea. It's a great question. David is asking about, do we look at a multi-scale notion of symmetry so that we're capturing longer range symmetries and not just the greedy symmetries that arise from a medial axis transform, right? And there's a whole community of work that began in the 80s by Tony Lindbergh at KTH that looked at multi-scale blobs and ridges, all of which are symmetric representations. We know that the multi-scale blobs and ridges gave David Lowe the anchor for what he did with SIFT, for example. But that community has thrived for many, many years. So I absolutely agree that we need a way to come up with a multi-scale representation of symmetry in the image. There are local symmetries at coarse scales. There are local symmetries at fine scales. We see that scale clearly in the top 50% gives you better performance, suggests to me that symmetries at a coarser scale may be more salient in the image than symmetries at a fine scale. So we need some way. Maybe the classical medial axis-based technique is insufficient for that. I don't know, right? The medial axis does not naturally give you a coarse-defined medial axis. But there have been people that have worked on that over the years. So it's possible, right? But it's combinatorial complex. Which contours do you group together, right? It's a very challenging problem. It's a great question.
Thanks. A few, with more questions. So yeah, Robin, Esther, over here, and then.
I was just wondering if maybe the separation for the symmetries, at least for the CNN, might just be the receptive field. Like it's much harder for it to, at many layers, it would have to, go many layers down for one pixel to see, symmetry across the image or something like that. So just by having it from the get-go.
Yeah, but the separation helped us. Larger separation helped us in the network, right?
Oh, I thought that was when it was passed to the network.
Yes.
Yeah, so if you give it to the network,
I see.
then it doesn't need to find the large scale ones. It's just, I'm just wondering if it's like a receptive field thing or an importance of the future.
Good question. I can't tell you that. Yeah, it could be. Yeah. That's a good question.
Just to build off of David's question earlier, you mentioned how at the start of the presentation, you mentioned how a lot of this is based on grouping. I was wondering if you guys have tried using like vision transformers, which have shown to do like visual grouping of symmetries before.
No, we haven't tried that.
Is that something that you might like?
Yeah. If you could point me to something that you think has begun to explore that, that would be great. Yeah.
Do Esther and then Karen.
Do you have any thoughts on how like other visual systems, not the human ones, leverage symmetry, or if it's even leveraged at all? Because like if I'm looking at visual systems across the board for organisms, humans don't necessarily have the best visual systems, and they definitely don't have the best in like any specific like regard. So I don't know if like we've looked into other visual systems and seeing how they process their data or like how they make their data in some form.
Yeah, it's a great question. Do other organisms, visual systems, process symmetry similarly or differently?
Yeah.
Do the tasks that they have evolved to survive on make use of symmetry somehow? I don't know, but I can tell you that my close collaborator, Zygmunt Pislow, has the most encyclopedic knowledge of vision systems across all organisms. If you contact me, I will get you that answer.
There was a study by Eleanor Roche where she went to some uncontacted tribe who had no representations of any types of shapes.
Yes.
She gave them different shapes to remember, and I think the ones that they remember the best were the regular polygons. And so I guess it's learning that, so that type of proclivity and salience to symmetry, is there any kind of way to quantify like the informational content of symmetry? And like, for example, even if you flash different shapes, you could remember them better. So maybe that's something that what the networks are doing is a byproduct of kind of maximizing how much information is captured.
Yes, it's a great question. One thing we are looking at is if you compute symmetries across the entire scene, can we use saliency scores as a mechanism for visual attention to draw your attention to particular regions in the image where there's high symmetry, which is what you're getting at. Maybe high symmetry and attention correlates with memorability of these kinds of scenes, right? So that's something we're definitely going to look at.
Okay.
Right? There's two steps for this closure problem. The first step is getting to the closure, where you fixate somewhere inside the closed object, right? Then the question is, how do you compute the closure once you're in the vicinity of that object? What I talked about today is only about computing the closure once you're looking at the object. Once you're foveating on the object. You certainly can't compute that closure in the periphery, right? I mean, Aaron Hertzman gave a great talk here a few weeks ago about foveal vision and how if you're fixating on a particular region, it's hard to measure the distortion in the periphery of kinds of shapes, right? So if you rather take a large image and map it into different regions where you're attending to them individually, that gives you a photograph that more mimics what you would see in painting the world if you tend to look around, right? So I think symmetry is a great mechanism for visual attention. That's something that we want to try. I think memorability, symmetry gives you more simplistic, compact representations of shapes, right? There's great work by Michael Layton that talks about how any complex shape can be broken down into a series of asymmetries inserted into the object. Everything reducing either to a square or a circle in the extreme case, but you can take any shape and introduce asymmetries into it and yield whatever shape you want. So symmetry really is a mechanism that captures complexity of a shape, and maybe that explains why Roche's subjects remember the simple symmetric shapes and polygons more than the very irregular looking shapes.
Oh, we've got a couple more. I think Babak, Avia, and then was there the question back here? Did you? Oh, okay. Well, we'll keep going just a little bit longer. And okay, so let's, Babak.
Babak.
Hi Sven, great talk.
Hi Babak, thank you.
It may seem a little bit tangential, but it's related. Are you aware if there are any brain impairments, any pathologies that influence or affect the perception of visual symmetry? Are there people who lack the perception of symmetry?
Again, I don't know, Babak, but I'm going to go to the same source that I mentioned for your question, and I will get you that answer, I promise.
Thank you.
Yeah, you're talking about people that have lesions or whatever in parts of the brain where somehow they've lost the ability to attend to symmetry, non-accidental symmetry. Yes, I don't know, unfortunately.
Thanks.
Javier?
Yeah. So you talked about the development of the human visual system and the type of training data that we see, and we develop in the 3D world, right? And so we interact with the 3D world. And so I'm wondering if you gave thought to 3D symmetries and whether or not so maybe we look at an image and we are able to kind of infer the 3D of that image, and if that pixel will have symmetries in 3D but not in the image, we will score that pixel high or something of that sort.
Yes, so Aviad is asking about what is the relation between symmetry in 3D, which is of the world that we're evolving to look at, and the kinds of symmetries in 2D that I've been looking at now. Let me go back, Aviad, just quickly to Biederman. So first of all, I agree with you. We have evolved to detect the symmetries in the 3D world. And I personally believe that since symmetry is highly viewpoint invariant, we have learned to infer 3D symmetry from 2D symmetry. That's my own belief, right? Much of my work has been based on that over the years. That goes back to my thesis, in fact, so long ago. That's exactly what Biederman was suggesting here. Biederman looked at 2D symmetries, both ribbon symmetry and taper symmetry. He used that to infer three-dimensional generalized cylinder properties, right? Is the cross-section sweep function constant? Is it increasing or is it increasing and decreasing? So it's one example, Aviad, of a body of work, very influential body of work, that felt that we're using the viewpoint invariance of 2D symmetry to infer the 3D symmetry of local 3D symmetric parts, which, as Mara Nishihara showed, when you compose them together, you form objects that human subjects can recognize very quickly and effortlessly. Right? So, if you're asking me, Avi, I completely agree. I think that all of this 2D analysis, and that's the reason I look at local symmetry, because it's the local symmetry that's viewpoint invariant, not the global symmetry. Global symmetry that's viewpoint invariant is accidental. Just so happens that all the flowers in ImageNet, because they're taken by photographers to look right at the front of the flower, that's not how you see flowers most of the time, right? That global symmetry is disrupted. But what you get is the local symmetry. So how do you infer the 3D symmetry from the local symmetry? And in fact, the reality of these medial axis transforms is that they're not particularly good. They're not particularly good at inferring 3D medial axes from 2D medial axes, especially in the presence of self-occlusion. Right? So they may not be the best way to infer 3D symmetry, but Aviad, I would say that's a really important goal of 2D vision. You can use non-accidental 2D symmetry to draw your attention to two pieces of the world that you are sure belong together, because the probability that they belong to different objects is extremely low. Once you've locked in on that symmetry, then the question is, what can you infer about the 3D shape from that local symmetry? Can you reason about the other surfaces that might be adjacent? Right? And I talked about that, my hypothesis for why symmetry is important, because we're trying to look at the relations among surfaces in the world to infer 3D properties of the shape. And those surfaces project to symmetries. And what you have in Biedermann's theory is you're looking for ellipses that are adjacent to symmetry sections. And from that, you infer that it's a circular cross-section that sweeps either a parallel constant cross-section shape or an increasing shape and so forth. So Aviad, I hope that gets to your question, that I think the link between 2D and 3D symmetry is extremely important, that we see the world in 3D, not in 2D. We evolved to see the world in 3D, not in 2D. Vision is not about looking at images. It's about looking at the 3D world. Right?
Yeah.
That's my strong belief. I can't prove any of this, of course, but that's what I have evolved to learn to believe myself, and that drives much of my work, even though what I'm focused here on is just that first shallow level of finding 2D symmetry and figuring out what it can do to help understand the 3D world.
Yeah, that does answer my question. I was thinking in the other direction, actually, because you were talking about inferring the 3D symmetry from the 2D symmetry, but it could be that the direction is we infer the 3D world from the 2D image and then figure out the 3D symmetry and project it back to the 2D. So like going in the other direction. But
Absolutely.
yeah, that's really interesting.
So I would agree with you, Aviad. Biedermann, for example, believed that you locally recover the 3D world. It's not just a complete bottom-up layer of the entire 3D reconstruction. You have local symmetric evidence in the world. You infer local 3D symmetric parts. Then you reason in 3D about their attachments. And then you index into your database with small collections, as you can see here, Biedermann, small collections of 2 or 3 symmetric parts and their relations and their attachment locations, attachment angles, Mara Nishihara. This minimal information is enough to quickly queue or do primal indexing, as they call, into a database of 3D shapes, right? So I guess the big point, Aviad, is you're definitely using 2D to infer 3D. I would argue it's not at a global level, it's at a local level. Symmetry can really help you find local pieces with which you can invoke top-down knowledge, prior knowledge, to help fill in the rest of it, depending on the task, right? If your task is just to navigate this world and you need fine metric 3D shape, then maybe this isn't so important, right?
So how sensitive are we to projection then that like might break parallelism or other symmetries?
So you can do something I did 35 years ago. In fact, you can take a 3D symmetric part. You can tessellate a viewing sphere. You can look at all possible viewpoints of that 3D part. You can measure symmetry in your views. You can tessellate the sphere at different resolutions. You can model local feature extraction operators, right, and build that into your model. How much blurring, how much confusion do you have locally, right? And you can measure the probability of a so-called degenerate view where you lose the symmetry, right? Biederman showed examples where you have a blender, you show the top view of the blender, right? You don't see, it's hard to recognize it as a blender. It's like a circle inside a square. Another example is if I show you a cylinder from the end, all you see is a circle. You might infer that that's a sphere or the cone facing me. There could be all kinds of interpretations, right? You've lost one of the key symmetry properties that defines the cross-section sweep function of that part. So you can try to model what's the probability of taking an object in the world and actually viewing it degenerately. You can also find a strong correlation between degenerate views and losing particular axes of these symmetric parts.
Okay, maybe one last question.
Michael.
Hi, thanks for the talk. Thinking about the experiment where you take out the corners, you take out the middle parts of the segments.
Right.
So one way in which I was thinking about it is maybe the information is just not there when you can't do the task successfully. You've taken out the information. Another way of thinking about it is the information is there. It's just that it's computationally difficult to recover it. Maybe EGG mechanistically can do it, or maybe whether the trace.
Yeah.
Is there a way to delineate kind of those two possibilities?
I think so. Michael's asking if you look at the first set of experiments where we have middle versus junctions. It could be that you are trying to compute a closure, but it just takes longer, for example, but you might still come up with the right interpretation. So that is true, right?
An example, sir.
Yeah, I mean, I would believe that when you lose the middle segments, you increase the ambiguity. You have removed a powerful heuristic. So, for example, if I am at the end of a contour, and now I have to jump to another contour to close my circle, right? And the only evidence, the only relational information I have is something that is very close in proximity, like a junction. Turn right at this corner, 90 degrees or whatever, right? Then my search space, I would argue, is much greater, right? Because when I'm at a gap, I could go here, I could go here, I could go here. When you have the middle junction or the symmetry information, in making that local decision about how to close a contour, someone gives you a constraint that says, whatever you do locally, you're going to have to pass through this in your tour, in your cycle. And I believe that effectively constrains how you make that local decision. That's a hypothesis which we haven't proven, but we're hoping to get to that when we look at our closure study.
But that kind of says both, right?
Yes.
Maybe.
Yes, you're right. I agree. Yes.
Okay, great. Thanks for all the questions.
Yes, thank you. Great questions. And I got some homework for a couple of people. Yes.
Good. Well, let's thank Sven again.
Thank you all. Thanks again, David, for the kind invitation. Aviad and Marcus and all the people that joined online and came today. Thanks so much. It's a lot of fun sharing this work, and maybe down the road I'll have some more results to share with you all. Thanks again for coming.